Okay everyone, let us get started for today's class. Welcome to the week 10 lecture on Introduction to Developmental Biology, PMRF-NPTEL session. As you would know that these are problem solving sessions that are done by the NPTEL in collaboration with uh, PMRF, and so in these particular sessions we go through the different quizzes of the previous year's run of the course so that we get a good idea of what the course is about, the concepts behind the course, and help us answer different questions of this year's run of the course. So today is the 10th week and we are going to finish up our discussion on plant development and then follow it up with introduction to early mammalian development. So first I would like to quickly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur and I am taking this course. So how we do this course is that I will just show you a question from the previous year's run of this course and give you a bit of time to try to figure it out, and then show you the concepts behind the question and then answer the question so that we learn together. Okay, so we start off with a question on plant development. So the question is, in the image shown right here, which part will eventually give rise to organs involved in fertilization? So if you have been following the previous week's material, you would know that this is a developing plant embryo, and there are different parts to it which are labeled right here, and the question asks us which of these parts are involved in forming organs that this plant after it grows up will use for fertilization. So which part of this embryo will give rise to different parts that are required for fertilization in the adult plant? So we have four options, A, B, C, D. I will give you a moment to figure this out and answer this. Okay, let us look at it. So there are different parts to the growing plant embryo at this stage, and all of these are marked right here. We'll go from the bottom. So as you would remember that this part, this brown cells right here, it is known as the suspensor. It is not part of the embryo itself, but it is like extra embryonic tissue which helps in providing food supply to the embryo via the endosperm. So it kind of acts like a placenta but inside the seed. Followed by this green part right here, which is known as the root meristem. So meristematic tissues are cluster of stem cells which give rise to the entire root and shoot, which comes from the shoot meristem right here of the plant. So these stem cells will give rise to essentially the entire plant in both direction, top and bottom. Followed by we have this yellow tissue right here, which is known as the procambium. This layer will give rise to the vascular tissues in the adult plant with the xylem and phloem present in the innermost region. Followed by the ground tissue, which is the region between the vascular system and the external most region, which will be formed by cells right here, which are known as the which is the dermal, essentially the bark. And finally we have these two heart shaped structures. So the entire thing looks like a heart, the edges of the heart. This will give rise to the cotyledons. So these aid in initiating photosynthesis. So essentially the seed's original leaves that helps in initial photosynthesis. So yeah, cotyledons aid nourishing by initiating photosynthesis before the adult leaves form from the shoot meristem. In some plants, they function as a food reserve in addition to the endosperm. The meristems, which are the shoot and the root meristems, are a cluster of stem cells, and the entire plant body, the sporophyte body, will be formed from this meristem. and people have been looking at different mutants of all of these to figure out what exactly is their function. And a lot of study has been devoted in Arabidopsis, and using different mutants they have been able to figure out that the shoot and the root meristems actually develop independently. So if you are a shoot meristemless or hobbit or a topless mutant, all of these particularly inhibit independently either of the meristems, so that particular parts of the meristem do not form. So you can have a topless plant which doesn't have a shoot meristem, and thus you do not have anything above the ground, but the roots do form. So all of this has been done and pretty figured out. And if we go back to our question, which of these structures in the embryo will actually give rise to our parts required for fertilization, which is all obviously going to be the flowering parts, and that will obviously come from the shoot meristem, the green top part right here, and not obviously from the root meristem which goes underground. So to answer a question, in the image shown, which part will eventually give rise to organs involved in fertilization, it is going to be B, the shoot meristem. Okay, we move on to the next question. The plant hormone blank is important for initiating dormancy, whereas the plant hormone blank is required to break dormancy. So this we will discuss in a couple of slides, that once the seed is enveloped and like the seed entire seed is formed, it needs to be propagated far away from the original tree. So that is what the fruits are for. That is how some seeds actually get attached to the skin of or fur of different animals by hooks and claws, so that they can be propagated far away from the original tree. And during this time, say when a particular fruit eating bird has eaten the fruit, so that the seeds now is in their digestive tract, and then wherever the bird will go next, probably far away from the original tree, the seeds will come out and then give rise to the new plant. But during this time, when the seed is on the tree itself or being propagated by any way, it cannot start germinating, because obviously if it starts germinating, it is not going to find a proper food source or the environment to form a new plant. So the original seed actually lays in a period of dormancy till it can reach a suitable environment for germination and forming the new plant. So to maintain it in dormancy, there is a particular hormone that is majorly involved in maintaining that dormant period. And then after the plant has figured out that it is in a suitable environment to germinate, then there is a second hormone which breaks the dormancy and initiates germination. So we are being asked what the two hormones are, A and B. So I will give you a moment to figure this out if you know this. Okay, let us look at it. So as we are talking about dormancy, once the basic body plan is laid, essentially the embryo has reached a particular stage, there is a shift to creating the food reserve. Genes encoding seed storage proteins are abundantly expressed after food accumulation, Metabolism slows down, connection to the ovary in the original mother plant is cut, the seed desiccates. The seed throws out a lot of water so that it can remain dormant at a very low rate of metabolism, so it doesn't use up all the food resources and can stay dormant for a longer time. And integuments, which are the external surface of the seed, harden into the seed coat. All this process entering into the dormancy is a result of a precisely regulated genetic program, after which they become dormant. And for example, we here have an example of a viviparous maize mutant. So maize, the corn that we have seen all around us, the seeds are originally desiccated and lay dormant, they do not germinate. And so you have the corn looking, as we see that the seeds are just stuck here as hard, uh, with a hard seed coat and are hard to touch. But if you have a viviparous maize mutant in which the plant without the seeds, without even finding a suitable environment to germinate, start doing so right in the uh, stain call, instead of being first finding a suitable environment, it starts germinating right there on the corn cob. Then you have things like this. You can see the seed has started to germinate and form the shoot meristem, and this is very bad for the seed because it is obviously not going to find suitable resources to form an entire plant right there on the corn cob. It has actually first to at least reach the ground around it. And this is what happens in viviparous maize mutants, where the the change from dormancy to germination occurs really fast, and there is actually no dormant period of the seed, which is actually required for the seed to germinate into a full plant. And what hormones are essential for this? The hormones that are essential for this are first abscisic acid, which maintain dormancy. So this hormone is released by the seed so that it maintains itself in a dormant condition. And later on, when it has been prepared to germinate, it releases gibberellins, which promote breaking dormancy and germinating. So dormancy is maintained by abscisic acid, and then dormancy to actually starting up the metabolism again, preparing to germinate. All of this, which is breaking dormancy, is done by the gibberellins. Okay, and yeah, this is not this we deal with later. So the plant hormone A, which maintains dormancy, is abscisic acid. Plant hormone B, which breaks dormancy, is called gibberellin. Okay, moving on. Nope. Okay, going on to the next question. So now, after our plant has germinated, we are looking at development of the plant to an adult plant. So the question here is: scientists have recently discovered a plant species that will bloom flowers only after surviving a winter period. Which of the following pathways may be dominant in the flowering pathway in this plant? The options are gibberellin pathway, autonomous pathway, photoperiodic pathway, vernalization pathway, or auxotropic pathway. So some plants, if you have seen palm trees or banana trees even, they do not, uh, the moment they form the adult plant, they do not start flowering immediately. Many plants actually flower at a really, like after one season is what they say, that you have to grow the plant for one entire season, and in the next season the plant will actually flower. So all of this is maintained by a lot of pathways, and we are being asked if a particular plant will only bloom after surviving one winter period. So if it has not seen a winter, then the plant will not germinate a flower. So what do you think is the pathway that controls this? So five options, only one is correct. I will give you a moment to figure this out. Okay, let us look at it. So, interestingly, as we have seen in earlier discussion on vertebrate fertilization, not vertebrate, animal fertilization, such as in case of Drosophila, we have seen that the pole cells, which are the cells which will give rise to the germline, are segregated early in during development, like really early, and are kept aside away from every other somatic cells. But in case of plants, no such thing happens. So the germline is not separated from the plant, but instead the plant can decide to actually have any of its cells start behaving like a germline, so giving rise to our different male and female reproductive parts in case of plants. So it is not specialized cells that do it, but cells at a later time point can give rise to these particular reproductive organs. And these reproductive organs actually depend, like how many seeds the plant is planning to form depends on a lot of things. So obviously a plant, the fruits that it will form, the endosperm that it will form, how many seeds it will form, will actually depend on what the resources the mother plant has on hand. So there is an optimum balance between how many fruits it can bear and how many seeds it can bear. So it can't just go ahead and form really low quality endosperm and fruit and a lot of seeds to go along with it. It has to maintain a balance, being there being a correct number of seeds to have a very optimum endosperm for the seeds to be propagated very well. And this obviously is very well regulated, and the plant is able to sense information from the environment around it and determine how many seeds and fruits it should form. So there is an entire growth period that some species of plants do. Some plants actually initially only grow up to a juvenile vegetative growth, in which they have formed something looking like an adult body, but this adult plant adult this plant will still not give rise to reproductive organs or seeds. It has to undergo changes to form an adult vegetative body. So there is a time period between growing from juvenile to adult. This happens in plants which live for more than one season, not annual plants which grow and then die just after one season. So this the plants have to go from juvenile to adult stage, and then from the adult vegetative growth it has to go into inflorescence growth, that it is trying now to make flowers, to actually form seeds at a later point, and it is guided by a lot of things. One of them being the photoperiodic pathway. So obviously we see most flowers in the springtime only, because that is the optimum time when flowers can be made, and then you start fertilization of flowers from different pollens and stuff. And so there is plants actually can sense this, they sense how the length of the day growing or the length of the day shortening, and this is the photoperiodic pathway. So obviously once the length of the day starts growing, that is when they will have flowers, because they do not want to have their seeds being propagated in dead of winter when they will obviously die. So they sense the length of the day to determine that yes, the summer is approaching, now is the time, or the winter has passed, now is the time when I should form seeds. Autonomous pathway, which is pre-programmed into the plant to form flowers at this time. Vernalization pathway, which is a pathway where the plant needs to undergo some kind of external harsh stimuli, such as a plant after surviving a winter season, it is now going to start forming flowers. This and the winter season is hard on the plant, but after it has passed, now the plant is ready to bear flowers, because the plant knows that yes, I cannot form seeds during the winter, but once it has passed and it has seen one entire season of winter, then it can actually form seeds. So that depends on harsh external stimuli, which is the vernalization pathway. And finally, gibberellins again play a role in formation of seeds, or for formation of flowers. So all of these together give rise to floral meristem identity genes. which determine which location where the flowers will be formed, and then the second order of genes, the floral organ identity genes, actually give rise to the flowers, and you have a flower being just there. So this is how the entire pathway works. So scientists have discovered a plant species that will bloom flowers only after surviving a winter period. Which of the following pathways may be dominant flowering pathway in the plant? It is going to be the vernalization pathway, because harsh external environmental stimuli which depends on temperature. Okay, moving on. Which of the following tissues are responsible for increasing the stem girth? So size of the shoot, which of the or size of the main bulk of the plant, which of these are responsible for increasing that? And the options are cork cambium, vascular cambium, intercalary meristem, shoot apical meristem, and root apical meristem. So here we are not talking about height of the plant, but rather the width of the plant. And this is more than one option correct kind of a question, so you can choose more than one. I will give you a moment to figure this out. Okay, let us look at it. So again, we are going to talk about early development in the plant. So as we saw that there are two types of meristems, the root meristem and the shoot meristem. When lateral roots grow out of the root meristem, that is, obviously there are going to be a primary root, and there are multiple other roots that we have on no plants have. So when a lateral root grows out, the entirety of uh, the tissues that contribute towards the lateral root come from the precambium or procambium, as well as the other layers as well, the ground as well as the dermal. So the root actually is connected to the procambium, which will later on form our plant's vascular system. So as you can see there, the lateral root has all the three layers that are required, and towards the end where the root is actually growing, so there is going to be a root cap. This is the part that is burrowing inside the into the soil, and this degrades off real fast because cells die off by their activity to go burrow deeper and deeper. And there is this part just above it, which is known as the apical meristem, and this is a source of stem cells that keep on making more and more cells to contribute towards the root cap. And since they are stem cells, they do it as long as required to form more and more cells to form the root cap, which then fall away, and then more cells are added. The shoot meristem, on the other hand, so if there needs to be some structures coming out of the shoot, so such as the leaf, then the layers which contribute to it only are our ground tissue and the external dermal layer. The procambium does not contribute towards growth of our leaf primordia. So, and then we have an apical meristem, which contributes towards stem cells for growing towards the top. So this is how all of this forms. And which of the following tissues are responsible for the stem girth? So the thickness of the entire uh, stem. So as you can see right here, majority of the thickness is contributed by the vascular tissue, that will be formed by the procambium, and or the vascular uh, cambium as well as the ground tissue. The dermal does not contribute towards the thickness because the dermal is only going to be the final bark on the external of the tree, which does not contribute much to the width. It is mostly going to be the ground tissue and the procambium, which gives rise to the cork cambium and the vascular cambium. Anything else, the intercalary meristem, shoot and root apical meristem, it gives rise to growth in the top and bottom direction rather than increasing the girth. Okay, going on to the next question, we are coming back to formation of flowers, and on the left we have genes which are known to be involved in growth of flowers but at different time points, and on the right we have what the classification of the genes are: the region specifying genes, the vegetal meristem to inflorescence meristem transition, the floral meristem identity genes, floral induction gene, and floral organ identity gene. So we have to connect all of these genes to their classification. It is match the following. So I'll give you a minute to figure this out. Okay, let us look at all of this. So as I said that we actually the signal for the plant to form the flowers, it can be directed from multiple different pathways: the photoperiodic pathway, the vernalization pathway, the gibberellin pathway. Here we are going to look at in the photoperiodic pathway playing a major role. So obviously, as I said, the plant can figure out the days getting longer by the amount of sunshine they're getting, and one of the proteins which actually can receive the signal and interpret it is known as CO or constants. So constants, and then it transfers the signal to the flowering locus T or FT, and both of these are known as flower induction genes because they start the entire process. FT gets transferred. FT gets transported towards the apical region of the plant, where there are other genes such as the terminal flower 1, which inhibit genes such as flowers. So flowers, what it does is that it prevents growing of flowers at a particular region. So once the FT signal reaches this place, TFL1 will start inhibiting flowers to induce transition from a vegetative state to inflorescence state, so that the plant is now primed to form a flower. So transition of a vegetative meristem to an inflorescence meristem occurs by terminal flower 1, and once this happens, the signal from FT as well as FD, which is another gene involved in this, is transmitted to the floral meristem identity genes. So these genes identify where the floral meristem will form, so the stem cells which will give rise to the flower. And after that we have the floral organ identity genes, which will actually form the flower. So the floral meristem genes, two floral meristem identity genes. So the where the floral meristem is formed is going to be determined by the floral meristem identity genes, which are leafy, apitella, one, or cauliflower. So let us look at these. So this is a normal plant with flowers, as you can see from the sides. And in case of a leafy mutant, you do not have any flowers, you only have leaves. In apitella 1 mutant, you realize that there are no sepals and petals. So these sepals and petals right here are not formed, and these are just the anthers and the stamen being formed. And in case of a double mutant, you have essentially nothing getting formed here. So instead of there being flowers, there are only leaves all around. It is all green, and even the stamen and the petal is getting transformed into vegetative organs. And if you have a double mutant with apitella AP1, so in this case cauliflower and AP1 mutants, so this still stays in, so it tries to form a floral meristem, but it is vegetative in nature and looks really like a cauliflower. That is why this mutant is known as the cauliflower mutant. So essentially, after the photoperiod and autonomous pathways determine that yes, this is a good time to form flowers, the first guys who start the process off are the flower induction genes, which as we saw are FL1, it is FT and CO, which are the flower induction genes. The signal is passed on to the next guys, which are our TFL1, which are the change the vegetative meristem to a inflorescence meristem. Next in the line are floral meristem genes, leafy, apitella 1, cauliflower, which change the which determine that yes, we are now going to form a floral meristem and identify where exactly the floral meristem is going to form. So essentially this is the precursor to the flowers. Then we have region specifying genes, which specify which part of the flower forms where, such as Superman. And finally organ identity genes such as AP2, agonymous, AP3. These guys we are going to look at later. So constants, as we have seen, is a floral induction gene. So one goes to D. Terminal flower 1, TF1, is going to be our vegetative meristem to inflorescence meristem transition gene. So 2 goes to B. Apitella 1 is going to be meristem identity gene. 3 goes to C. Superman, we just saw, is a region specifying gene. So 4 goes to A. And AP3 is our floral organ identity gene, which is 5 goes to E. So the second option is the correct one. Okay. Yes, so this is a very interesting thing that happens in plants. So now we are going to talk about our floral organ identity gene. So where is our sepal getting formed, petal getting formed, stamen getting formed, everything. And we have figured this out very well. And this is known as the ABC class of proteins that do it. So what is the ABC class of proteins? So there are 3 different A, B and C proteins. When there is only the A protein gene being expressed, that structure forms sepals. When A and B both are expressed, they form the petal. C and B are expressed, it is forming the stamen, and only C forms the carpel. So C forms carpel, A forms sepal, and A and B combination forms petal, and B and C combination forms stamen. And this is how essentially this is like a homeotic gene scenario, as we saw that homeotic genes can form different structures in animal embryos. Similarly, it happens in case of plants, that this class of ABC proteins are also homeotic genes which determine which organ forms where. So now scientists discovered a mutant that does not form petals, but can form sepals. So sepal are forming, petal are not. What are the possible expression patterns of ABC class proteins in this mutant? So here we have different patterns and we have to choose the ones where sepals form but petals do not. And this is more than one option kind of a question, so go ahead and choose more than one option which you think is the correct ones. Okay, let us look at this. Okay, so wild type and mutant phenotypes of Arabidopsis class A, B and C floral identities. So we have already seen this. So G, wherever the gene A is expressed, it always forms a sepal. Wherever A and B both are expressed, it forms the petal. B and C both form the stamen and C only forms the carpel. So we have examples of different all of these genes being expressed. So in case of AP2 mutant where A is not there, everything becomes C, and C, as we have seen, forms carpel, and where it is along with B it forms stamen, and where it is alone it forms carpel. So something like a situation like this, you have the stamens and the carpel forming but nothing else around, which is the sepal and the petals. In case of a B minus mutation, so you only have A which forms sepal and carpel, no stamen being formed or petal being formed. So this is how it looks. You can see right here, carpel and sepals, no petals or the stamens. And finally C mutant, you get rid of the carpels. So you have sepal, petal and sepal. And you can see that it is a very nice looking flower with all the petals around it, but where the stamen should have been and the carpel should have been is completely empty. The actual reproductive parts of the flower are missing. So this is how the ABC system works. And our question is, our mutant can form petals, but cannot form petals, but still forms sepals. So petal, if I want to remove petals, so I will have to remove this category, but I still have sepals. So A is being expressed. It is probably that B is just not being expressed. So I just need this to have not, our plant probably does not have a A and B combination, but can have any one of the others. So the first option, so there is no AB combination, so no petals, but sepals are getting formed. So this is correct. This is also correct because there is no AB option, but there is a. Here we have a AB option which will give rise to petals, so this cannot be our one. Again, AB option, our still has petals. And finally, this one doesn't have A in it, so it does not even form sepals. So A is required for sepals. So there is no A here, so no sepals. So this cannot also be our option. So only option correct are A, the first two. Nice. Oh, moving on. Now we take a break from plant development, and now we move on to early mammalian development. We start off with a very straightforward question. What are the major reasons mammalian embryos develop within the parent organism as opposed to eggs in reptiles and other animals? The options are Development of specialized features requires a constant high nutrient supply, which is given by the mother via the placenta. Better protection of embryo within the parent organism as opposed to in an egg. Mammals are warm-blooded animals and hence require a constant supply of blood from the parent during development. This is a very important one. And mammals invest more resources into improving the survival of few embryos, while reptiles invest more resources into increasing the number of embryos produced. This is also true, as you know, that in case of humans, most of the time it is only going to be one embryo that grows out in one gestation period, whereas in case of reptiles, you have multiple eggs being laid, and it is just quantity over quality rather than the other way around in case of mammals. So I will just give you the correct option. All of them are correct and all of them are valid reasons why mammalian embryos grow inside the mother instead of outside in an egg. So specialized features require constant high nutrient supply. So mammals do have a lot of specialized features and are pretty much at the top of the evolutionary hierarchy. So we have a lot of things that needs to be formed, and this all of these require high nutrients to do, and this cannot be just the nutrients being available in the egg. Better protection of the embryo within the parent organism as opposed to in an egg makes sense. Pretty straightforward. Mammals are warm-blooded animals, so obviously in case of this one is out there, I guess. This one I can imagine arguments on the other side, like in case of avians, they are also warm-blooded, but they still have eggs. So I don't know about this one. So mammals invest more resources into improving the survival of few embryos, while reptiles invest more resources into increasing the number of embryos produced. This is also true. Okay. So we actually go on to discussing about human embryonic development, and the first question that we ask is, how many cells are present in a human embryo at the morula stage, which is a very early stage during embryo development. So you guys can take a try at this. Okay, let's look at this, and here I have a video to show you, teach you about early embryonic development in mammals. So I will go and share my screen to show you a video of this. Not able to share. Should be it. Okay, so this I will this is a video by Khan Academy, which very well tells you about embryonic development in a very simplistic manner. So I will just play this for you and you can have a watch.
We're going to talk about early embryogenesis. Say you're an egg cell, and you have this nice thick outer glycoprotein coat called the zona pellucida, and you've got your plasma membrane just inside of that. And a sperm has made its way through the zona pellucida and managed to get in through your plasma membrane and merged its genetic material with yours. You're now called a zygote, and you'd like to go on to form an embryo, but not much is going to happen when you're stuck as a single cell. And so what you've got to do is divide into multiple cells, and you've got to do it fast. In fact, you've got to do it so fast that you don't have time to grow. So you actually just split into 2 cells. And this process of splitting without growth is called cleavage. And you do this a number of times, dividing from 2 cells to 4, and from 4 cells to 8, all the while staying within the zona pellucida. So you've gone from 16 cells to 32 cells, and at this point you look different enough that somebody decides to give you a new name. So instead of being called a zygote, you're called a morula. And morula is just a Greek word that means mulberry, and you actually do look a little bit like a mulberry. So here's a picture of a mulberry from my front yard to prove it. This one's not quite ripe. So you're finished with the cleavage stage of early embryogenesis. You've gone from 1 cell and just divided without growing at all into 2, 4, 8, 16, and then 32 cells. And now more interesting things start to happen. So you're still stuck within that zona pellucida. So we'll draw it in here. And I'm not going to keep track of the number of cells anymore, I'm just going to draw them in. But what you'll notice is I'm drawing them in quite compacted. I'm drawing them all right in the middle of the structure here. And they do look like they're a little bit tighter together. And this is a process that's called compaction. The different cells within the morula start to get closer and closer together. And in fact, the cells start to get a little bit different from each other too. And you notice that these cells on the outside are a little bit different. I'll draw them in here a slightly different color. And we can tell them apart, and this process of being able to tell cells apart as they become different things is called differentiation. So here we have 2 separate populations of cells. This one on the outside, we'll call them trophoblasts, and this mass of cells on the inside we'll call embryoblasts. And so you're going to continue with your process. We'll move on to the next stage. Again, we'll draw our zona pellucida here, and we'll draw all of our trophoblasts along the outside here. Another interesting thing is happening to those cells in the middle. They're starting to clump even more. In fact, they clump so much that they all cluster at one end, leaving a little cavity on the other end. So here you can see your trophoblasts and your embryoblasts, this mass of cells in the middle. Some people actually call that the inner cell mass, and it's left you with this cavity, and that cavity is called a blastocele. Now, again, you are starting to look a little bit more different than you have in the past, so we're going to give you a new name. This structure that you've turned into with a blastocele and an outer cell ring of trophoblasts is called a blastocyst. In fact, we name this whole process after you, and after cleavage, the process is called blastulation. So also about this stage in blastulation, your zona pellucida starts to disintegrate away. Here I'm going to draw a little bites being taken out of it here as it falls away and disintegrates. And that's going to be important later on because you can't be stuck in this thing forever. And the next step in blastulation, you actually just lose it completely. So now we'll draw our trophoblasts completely naked without a zona pellucida. But more interesting things are happening to you. Your inner cell mass of embryoblasts is starting to look a little bit different. So you still have this rim on the outside here up at the end of your blastocyst, and you still have this mass here in the middle. But you've developed another cavity, and this cavity is called the amniotic cavity. And also, your inner cell mass of embryoblasts has started to differentiate more. And now it's got this layer on the bottom of it here, and the cells in this layer are called hypoblasts, while the cells in the layer just above it are called epiblasts. Now at this stage, we're pretty much completely free of our zona pellucida, and that's going to be really important for implantation. But that's a discussion for another time. We're going to focus on this ball of cells here. And I really want to stress that this is a ball of cells. It's not flat like we've got it drawn here on the computer screen. It's three-dimensional. It's spherical in nature. A spherical melon. So think of it like a melon. Here I'm going to draw a spherical melon. So now I want you to picture taking a big knife or a machete or something and just slicing the top off the melon like this. So here we'll erase the top part of our melon here, and we're left with a flat surface. So now that you've got a melon with a flat surface, picture yourself taking a pancake and just putting it right on top of this flat surface like that. And that's basically what we've got here with our blastocyst and this forming layer of epiblasts and hypoblasts. It's really a pancake. It's not two-dimensional here. It's a three-dimensional disc of tissue. So we're going to draw this again, but this time without the outer sphere of trophoblasts. But we're going to draw our pancake of epiblasts and hypoblasts. And what you can see here is our pancake has got two layers. So in fact, this is a very important structure in embryology called the bilaminar disc. And we're going to get another look at the bilaminar disc here. So I'll show you. We're actually going to draw a little plane through the disc here. And if we take a cut view through this plane that I've drawn, it looks a little bit like this. So this is still our bilaminar disc, but now we're looking at a slice through the bilaminar disc, as opposed to here we're looking at the entire bilaminar disc from the outside. So now we're looking at our pancake of our bilaminar disc here, and we notice we start to see something forming on the edge of it here. And that something looks like a little bit of streakiness that kind of splits our pancake right in two. You can think of it as pouring a little streak of syrup along the top of the pancake and dividing it into two halves. So here I'm going to draw it on the surface of the pancake, and then over here on our cut view you can see that this little streak happens right about here in the middle as it reaches the plane of our image. And this streak of syrup on our pancake has got a name. It's actually called the primitive streak. And the formation of the primitive streak marks the beginning of the next stage of early embryogenesis, and that's called gastrulation. Now what that primitive streak actually is just the site where the cells in this epiblast layer of our bilaminar disc start to migrate. Here I'll draw the paths of the migrating cells. They heap themselves up right at the primitive streak, and then they start burrowing their way down into this bilaminar disc. And they go out into the hypoblast layer, and they just kind of go all over the place here. But it all happens from that primitive streak. So as the cells migrate out from the primitive streak, we can see that our structure has changed a little bit. Now instead of having two layers, one layer of epiblasts and one layer of hypoblasts, all of those migrating cells have now differentiated even further, and we're left with a layer of cells on the top, a middle layer of cells as they go out into the body of the bilaminar disc, and we still have a lower layer of cells down here. And so now you can see, instead of the bilaminar disc that we actually have a trilaminar disc. So we have three layers, 1, 2, and 3, and these are actually called our germ layers. So we have a single layer on top, and instead of epiblasts, this layer is now known as ectoderm, and our middle layer is mesoderm, and our lower layer is endoderm. And cells from these three layers go off to do very important things, and each layer forms its own specific structures. So that process of the formation of the three layers is called gastrulation. Once our three layers are formed, we can move on to the next step in embryogenesis, and the next step is called neurulation. So we'll draw our three germ layers in. We have a layer of ectoderm on the top. In the middle, we have a layer of mesoderm. And then on the bottom, we have our endoderm. So this final stage in early embryogenesis is called neurulation. And as you might expect with neuro in the name, we're going to see some neural elements formed here. And now we have our trilaminar disc with our three germ layers. And in the middle of the mesoderm, the central layer here, we start to have further differentiation of cells. And right in the middle, we get this cord that starts forming. And I'm going to draw it as a little purple dot here, but what that really is underneath where the primitive streak was, mesoderm cells are differentiating into a cord structure, and that's called a notochord. Now in humans, the notochord doesn't go on to do a whole lot. It does form part of the intervertebral discs, and very rarely it will cause a tumor called a chordoma. But in general, its main goal and its main purpose is here in neurulation. Now, this differentiated bit of mesoderm actually induces a change in the ectoderm above it. So here we get a change within the ectoderm. I'll draw that in as kind of a thickening of the ectoderm right here. And that thickening in the ectoderm has a name. Since it's kind of plate-like when we look at it in section here, this is called the neural plate. Now we have our notochord formed and we have our neural plate formed. The next thing that happens, so we'll redraw our three layers here, our ectoderm, our mesoderm, and our endoderm. And here at the bottom we have our notochord forming within our mesoderm. And right above it, those neural plate cells actually start to dive into the mesoderm. And as those neural plate cells dive in, they start to form a ring structure. And actually, since this is a three-dimensional thing, this ring is more like a tube, so you can picture this tube going off into the rest of this pancake that we've drawn here. And as this neural plate zips up and dives down into the mesoderm, it becomes known as the neural tube. Now, this isn't a perfect process, and as the neural tube is zipping up from one end of our pancake to the other, little cells are breaking off from the ectoderm and going out into the mesoderm. Now, these cells actually have a very important role as well, and will go off and to differentiate into their own special tissues, and these are called neural crest cells. So once we have the formation of our neural tube and our neural crest cells are diving off into the tissues and differentiating, and we have a pretty good idea of what the ectoderm, mesoderm, and endoderm are, our early embryogenesis is complete. So just to recap, we started off as a zygote, a single cell that was fertilized by a sperm. Cleavage happened, we didn't grow, but we split into a whole bunch of cells and developed into a morula. The morula cells started to differentiate, and we developed trophoblasts on the outside and embryoblasts in the middle. We formed a little cavity and became known as a blastocyst. After the blastocyst cavity formed, or the blastocele, a second cavity called the amniotic cavity formed, and a pancake of cells across the sphere became apparent. Those cells continued to differentiate, and we formed a primitive streak. The epiblast cells dove into the primitive streak and started to differentiate into our three layers that became our ectoderm, mesoderm, and endoderm. And in the final stage of neurulation, we had development of a notochord, which induced a neural plate to form, and the neural plate dove into the mesoderm and formed the neural tube. Neural crest cells associated with the neural plate also went off into the tissues to further differentiate.
Okay, that should be that was a pretty good summarization of how exactly gastrulation, early embryonic development works, all the way from implantation to gastrulation. And now we can go back and continue our discussion on the question that we had. So this is what this is what all the stages look like in case of a human embryo. In case of a mouse embryo, so this is the 2-cell stage, 4-cell stage, uh, this is the 8-cell stage, and then we have compaction, and then we go on to have a 16-cell stage, which is known as the morula. So morula is when the cells cannot be, you can't, the cells appear as a ball of multiple cells. The entire structure appears like a ball of multiple cells, and it starts at the 16-cell stage but ends up with the 32-cell stage, where the ICM and the trophoblast separate. Then you have cavitation, which is known as the blastocele formation, and this is called a blastula, and after implantation it forms the blastocyst. So this is essentially the early development that the video also discussed. So how many cells are present in a human embryo at morula stage? It is six. It starts at 16, but the 32-cell stage can also be classified as the morula stage. So I guess 16 or 32, whatever you put in. Okay, which of the following embryonic parts prevents implantation of an embryo in the fallopian tube? The options are cell membrane, zona pellucida, blastocele, corona radiata, or the vitelline membrane. So I will give you a second to figure this out. Okay, let us look at it. So this is what the uh, how early fertilization works in humans. So this is the ovary where ovulation is taking place. Uh, the oocyte is developing with a coating around it, which is known as the corpus luteum, and it goes out of the ovary into the fallopian tubes. These fallopian tubes go all the way to the uterus. Just as the oocyte is coming out of the ovary, it is fertilized by the sperm, and after fertilization, the zygote starts dividing like crazy. We have the two cell stage, morula, and by the time it has reached the uterus wall, it embeds in the uterine wall as in the blastocyst stage. Now, interestingly, the uterus only has all the elements required for implantation as well as growth of the embryo, but implantation can also appear in this fallopian tube where the initial growth of the embryo occurs. And if implantation, which is the embryo sticking to the uterine wall and start growing, occurs here, then that is a really dangerous condition, because this just causes major can cause major damage to the fallopian tubes and the mother. And to prevent the embryo to implant early on in the fallopian tube, there is a particular layer that the embryo surrounded it in, which is known as the zona pellucida. The zona pellucida is like a membrane that we just saw in the video as well, which covers the entire embryo, so that it does not implant anywhere here, but only can implant here. And the reason why it can implant in the uterine wall is that by the stage, by the blastocyst stage, the embryo starts secreting various enzymes such as trypsin, which degrades the zona pellucida. So only at the blastocyst stage, after it has traveled all the way here, can it actually implant. So no fallopian pregnancy or tube pregnancy. So the zona pellucida prevents premature implantation. Upon reaching the uterus, the embryo digests a whole, possibly a trypsin-like protease secreted by the trophoblast in the zona pellucida, and comes out. So this is what it looks like. This is the zona pellucida, and the embryo is just breaking through. And here is where your uterine wall must be. And this is what it looks like here. So this is our trophoblast layer. This is our ICM, and here is the connection to our uterine wall, and so this is where the implantation is taking place. So endometrium is the layer where it gets attached. So the first attachment is via the ECM. So the ECM of the embryo and the ECM of the uterine wall interact, and after that happens, the digestion of the zona pellucida occurs, and then the embryo embeds deep in the uterine wall itself. So it degrades all of this and goes inside the uterine wall instead of sticking to the surface. This entire process is known as implantation. So which of the following embryonic parts prevents implantation of the embryo in the fallopian tube? The answer is zona pellucida. Okay, moving on. Which of the following are necessary to maintain stemness of the embryo? So stemness, it essentially means the pluripotency or the totipotency of the embryo, so that it can give rise to more and more cells. So stem cells, as you would know, are cells that can give rise to other cells, as well as have the ability to infinitely divide themselves. So these are obviously really important. So all our blastocyst cells are pluripotent. They can give rise to everything other than the extra embryonic tissue, such as the trophoblasts. And this is where our embryonic stem cells, that we hear a lot about in biology nowadays, come from. So which of the following genes are necessary to maintain the stemness of these cells? The option is OCT4, NANOG, CDX2, STAT3, or GATA6. This is obviously more than one option correct question, so I will give you a moment to figure this out. Okay, let us look at it. So this is what I was talking about. The blastocyst, as we saw, has two different parts: the inner cell mass and the trophoblast. The trophoblast gives rise to the cytotrophoblast, the syncytial trophoblast, the extra embryonic tissues that help in setting up the connection to the mother via the placenta. And the inner cell mass also has two parts: epiblast and hypoblast. The epiblast surrounds the yolk sac, and epiblast also can divide and form the amniotic ectoderm, which is the covers the amniotic cavity, which is essentially a liquid filled cavity to which acts as shock absorption for the growing embryo, and all of this is extra embryonic tissues. Now the epiblast itself gives rise to essentially the entire embryo. So the epiblast forms the embryo, but as you can see, the inner cell mass at this stage can essentially form the entire embryo, which is this top half. So it is known as pluripotent stem cells are derived from the inner cell mass that can give rise to all the tissues in the embryo. However, the blastocyst cells are known as totipotent. Totipotent means that it can form everything, so it can give rise to extra embryonic tissues as well, which the inner cell mass cannot. So that is why inner cell mass pluripotent, blastocyst totipotent. But where does this stemness or the stem cell characteristic come from? It comes from these genes. So here we have a very nice image showing the different parts. So here it is our inner cell mass, this is our hypoblast cells, this is our trophoblast cells surrounding the entire thing, and these cells are the ones we are talking about. So at the morula stage we divide into ICM and trophoblast. After the morula stage, the trophoblast activates the genes CDX2 and EOMase. CDX2 and EOMase actually, so these are differentiated cells now. Trophoblast cannot give rise to it. These are almost differentiated cells. These are not stem cells anymore. They have a particular function and cannot divide infinitely or form newer cell types. It can only like transition, so it can form newer cell types but cannot form infinitely give rise to more and more of the same cells. And to do that, CDX2 and EOMase have to shut down OCT4 and NANOG. OCT4 and NANOG actually are present in the inner cell mass along with STAT3, which continues to increase more and more like in a positive feedback loop, and these genes OCT4, NANOG and STAT3 actually maintain the stemness of the ICM cells, which later on give rise to epiblast and embryonic stem cells, everything else. And these cells in the ICM which have OCT4, NANOG and STAT3 are the embryonic stem cells that we hear so much about, that can give rise to any embryonic structures or any organs in the adult as well. And so ES cells are ICM derived cells expressing OCT4, STAT3 and NANOG. So if we go back to the question, which are the following necessary to maintain stemness of the embryo? OCT4, NANOG and STAT3. Okay, so that is all I had for you today. So thank you for joining in today, and I will catch everyone next week with the next week's lecture. If you have any questions, you can drop it into the NPTEL forum chat or the YouTube comments and we can discuss further. And that is all for my end for today. Thank you and see you next week. Thank you.